Min, thank you and all panelists for that very thorough primer on GCT in China and many important insights for all of us here in the States. Our next panel, also an international one, will be moderated by Dr. Lindsey Baden. Lindsey is the Director of Clinical Research at Brigham's Division for Infectious Disease, and he personally led one of the key trials for the deployment of the COVID vaccine. This panel will focus on the impact of the mRNA vaccines and lessons learned from the global purview of those vaccines. Thank you.
So welcome and thank you all for joining this panel. And you all have the bios of our three expert panelists, Kate, Melissa, and Ron. No need to introduce them. We will jump right in. And as we look back on what's occurred over the past year, and we've witnessed the potential of mRNA technology, its implications for addressing a global pathogen like SARS-CoV-2, and how countries like the UK and others responded to that threat. We now have to think about how we integrate and build on the lessons learned and move this technology forward and consider its potential for other diseases. So let me start with a question to you, Melissa. What are the advantages of mRNA technology for vaccines, and what are some of the downsides?
Well, I mean, clearly the advantage, the one clear advantage is just how efficacious they are. And with both the Pfizer and the Pfizer BioNTech and the Moderna vaccines, we're seeing 94 to 96% efficacy. I mean, that's just really remarkable. Another advantage, clear advantage, is just how very quickly we can manufacture the vaccines and how we can identify, once we know what protein to make a vaccine to, we can, within a day, design the vaccine and get it into manufacturing. And so it's a very fast process. Now, the current downsides, obviously, are the need to store them at low temperatures and their more limited shelf life in the refrigerator, and particularly at room temperature. And I know that's something that both companies as well as other mRNA companies are working on furiously. So hopefully that will, in the future, be less of a downside.
And do you see with cold chain that there are potential solutions to the cold chain challenge, Melissa?
Yes, I think that what you're seeing over time, since both Pfizer and we came out with our vaccines, we're already announcing extensions of the ability to store them for longer and longer times at refrigerated temperature. And so it's really one of those engineering challenges that we just need to keep working away at, and we'll get there.
I mean, I think, as you sort of alluded to, it was merely 15 months ago when this technology emerged as a potential response to SARS-CoV-2. And with the demonstration of efficacy, then solving many of these technological problems become worthwhile. And it is good to hear that the path to that is underway, and we've seen evidence as well in the deployment strategies. Sorry, go ahead, Melissa.
Well, I do want to correct something you said, Lindsey, and that is that I think the world became aware of the technology in the last year, but we've been working on this technology for well over 10 years. And we had our first in human vaccine back, and I'm looking over at my screen to just make sure I get the numbers right, but we had our first in human mRNA vaccine back in 2015, late 2015, early 2016. And so it's actually been multiple years, and we've now put many different vaccines into humans, including Zika, cytomegalovirus, RSV, to just name a few. And that's been going on for years. And so it's not that this is just a technology that came to bloom right overnight. There's years in the making.
Melissa, your point is very well taken, and I fall into the trap many of us fall into, becoming aware of this in the past year, where it really has been a technology under development for quite some time, with a variety of different antigens moved into human assessment over the last 5 to 10 years.
Yes.
So thank you for pointing that out. And that has also enabled us to better appreciate some of the safety features as well as the immunology. Taking a step forward into last year, Kate, what have we learned from the UK coronavirus experience and the vaccine response, particularly with the mRNA and the vaccine technologies?
Well, I would echo a lot of what Melissa said. I mean, we've learned that these are the quickest to manufacture and scale. And what we're seeing right now in India, the speed of vaccination globally is going to be critical to try and limit any absolute escape variants. So clearly we're seeing variants now, but I don't see any yet escape variants going beyond the current vaccines. But mRNA as a class or as a format is definitely the one that is probably the quickest to actually tweak and address any potential escape variants. And what I'd like to see, and you're right, I mean, it's the stability is the major issue, and probably is at the moment preventing widespread global distribution beyond the high and possibly middle income countries. So we have vaccinated about 50% of the 70% or so of the UK population that's been vaccinated has been vaccinated with mRNA, and the other half with the Oxford adeno. I guess what we've learned is we would like to be able to get it into a more stable form so that we don't have, even now, sort of the 2-week fridge temperature. And I have seen interesting emerging data taking, for example, mRNA formats and turning them into solids form, including some disposable, dissolvable implants. So things where you could potentially still get the mRNA immunogenicity, but with a single application, which doesn't necessarily need to be delivered by a healthcare professional. So it's the two needle-based deliveries with the cold chain that has been the challenge. And of course, we struck our initial deals in July last year. So our NHS teams had 5 months to plan for it, which is why it's rolled out smoothly.
And have there been differences in how the AZ chimp adeno rollout versus the mRNA rollout across the UK? Have there been important differences in how the vaccines are deployed that have impacted the delivery across the country?
Well, yes, I mean, AZ is an easier, more stable vaccine to deploy, but equally, and we also saw the allergy issue. So that's had certain consequences in terms of who can receive what. But broadly, there have been high vaccination centres that people have had invitations to go to, as well as local vaccination centres. And those who are the very frail elderly, the vaccines go to them. So the answer is it's been a good choice for people to basically do what or go to whichever centres work for them. But we have done it in very strict risk order. So being British, it's all been quite organised, being over 80s, and then healthcare workers, and then it comes down in age order. Obviously Moderna, we now have supplies for in the UK. So we are deploying 3 forms of vaccines at the moment. So 2 mRNA and the adeno.
Thank you. Given this technology and what we see what it has done in this past year, a novel pathogen emerges, to take a comment from a colleague, designing the vaccine is almost like ordering primers. And then for those who do PCR research, one knows the nucleic acid sequence and then can design the vaccine that is responsive to that sequence. And in the past year, we've been able to develop 2 different we being the larger community, Pfizer, Moderna, with partners. How does this technology then pivot to support other diseases? Ron, some thoughts on how we can leverage this technology for other kinds of illnesses?
Yeah, it's a great question, and to Melissa's point, I mean, the speed, the flexibility, and I think one thing that we can't forget about here is just the manufacturing aspect of messenger RNA here. 1.6 billion doses of vaccine have been produced in the last 12 months. And so that's something that I think would be an insurmountable challenge with just about any other platform. So as we think about going beyond vaccines and into other therapeutic areas, it's not inconceivable that we could start thinking about, first of all, any rare disease, for example, with a monogenic disease with a rare mutation, you could start thinking about putting that corrected messenger RNA into the right cell. So in the case of what we're working on, thinking about cystic fibrosis comprises somewhere on the order of about 2,000 different mutations. If you can actually put that corrected messenger RNA and deliver it to the lung and get it to where it needs to be, it can start to express that corrected protein. And so there's a lot of white space here for what we can start thinking about going beyond vaccines into other infectious disease areas, into other rare diseases, and even starting to think more broadly about oncology.
The ability to pivot for a sequence of interest, be it a genetic disease that is a host issue, or to a pathogen, which is what we've done in this past year. Melissa, can you tell us a little bit about how this prepares us for the next pathogen of concern and our flexibility to respond and respond rapidly?
Well, I think that, so first of all, I again want to just make the point that, yes, ultimately you can design an mRNA vaccine and it is like ordering primers, but a lot goes into that design. And because of the redundancy of the genetic code, you can't just pick any old sequence in the RNA to make a protein, because different versions make different amounts of protein. And so there's a lot of learnings that have gone into figuring out the design algorithms. But now that we have those, and I want to point out that we were so confident in our design algorithm that we literally made one mRNA, put it into GMP production, and that is mRNA 1273, our vaccine. And so you can actually now get to that point. But that we couldn't have done that had we not done many years of research into understanding the underlying engineering principles for the mRNAs. In terms of pivoting to new diseases, there's some really exciting things that are going on out there. Elon Musk has given CureVac a bunch of money to try to make mRNA vaccines very quickly. And there's also a program going on with DARPA called the DARPA Now project for nucleic acids available worldwide to try to, the idea would be that you could have a mobile manufacturing unit that could rapidly be deployed to anywhere in the world where there's a new outbreak of a disease. And then that manufacturing unit, well, first of all, you'd have to identify what the disease is or the virus, sequence it. But then once you have the sequence, very quickly make the mRNA in situ, so on site, and make a number of doses and get those to the local population in very short order. So I think that there's some really exciting and visionary and almost sci-fi ideas out there. But we're working on them, and I think in the next several years we're going to see real changes in how rapidly we can respond to emerging diseases.
I think one of the responses on that is also to see how to work with the regulators for that, because where you're taking a minor variant of an existing approved vaccine, I think there will be a very contracted regulatory process. The question is, how do you deal with?
Well, I think that's, yes. So it's often that the science and the technology outpaces the regulatory environment, and the ability of the regulators have to keep up. And so I think that's a really good question, and I don't have an answer to that now. I'm really much more the scientist than the technology builder. So I'll leave that to my regulatory colleagues.
But Kate, it's a good question, because as has been discussed many times, if you think about what we're using to deliver messenger RNA, these LNPs, I think if we have a robust, reproducible, safe, tolerable LNP, you're really just swapping out cargo. You're really swapping out open reading frame. And so from a regulatory perspective, we've actually, I think the last year has shown us really the flexibility and the speed and the insights that the regulatory authorities globally have had on this. And I think there is a lot of applicability that we can take from what just happened in the vaccine space over the last 12 months over to the therapeutic side. And as we start to think about families of genetic diseases, if you're using the same lipid nanoparticles to deliver this messenger RNA cargo, you could really start to think about really speeding up the delivery of these therapeutics in certain rare diseases and actually starting to address diseases that may go below the radar screen for large pharmaceutical companies because of the patient populations being so small. This becomes a very, very attractive opportunity for messenger RNA platforms.
I think that's a really good point, Ron. Thanks for picking that up, because the platform nature, the dose limiting toxicity is really around the delivery vehicle. And so once you have figured out what is doable with a particular delivery vehicle, then it's just a matter of swapping out to the mRNA sequence. And so, yes, that's absolutely a terrific point. And we can actually get down to what's now called N of 1, where you're making truly personalized medicines. We and others, we have a collaboration with Merck, and I know other companies are doing this too, of making personalized cancer vaccines, where we're really trying to make a vaccine just for one person to stimulate their immune system to fight their particular cancer. And so that's something that's already going on.
And I think that this speaks to, as the science advances to allow us to do personalized medicine where a therapy is designed for an individual patient, be it a genetic defect or a particular organism of interest. We see this with phage therapy for some of our resistant bacterial pathogens. Both the technology has to be designed to be able to go to scale, and the regulatory environment has to understand the platform versus the individual cargo. And the framework has to be created to allow this to advance rapidly. But it is the future of individualized medicine. And I think we in infectious diseases have dealt with this as we do antimicrobial susceptibility and try to treat an individual bacteria. But now we're moving it to an in vivo kind of delivery process. Very exciting technology. But we do need the regulatory side of the house as well as the scientific side of the house to come together with a path that makes sense.
And I think we may test some of that with these new emerging variants. So to the extent that, I mean, the approach we've got in the UK is to do mix and match or heterologous prime boost or heterologous boost, third boosts. And those studies are underway now. Is if we get an escape variant that is not responsive to the heterologous boost and evades the current vaccines, we will then obviously be needing to have tweaked vaccines. And that will be, I think, the first step to test the regulators to say how much data do you need to ensure the safety of these basically the new sequences in the same backbone. And I think we're starting to see from the regulators or hear from the regulators that it's going to be much more compressed. And if that's the case, that's already starting to address how they're going to regulate these new products.
Well, along those lines, Kate, and the question to you, Melissa, is the lipid nanoparticle, we have 100 micrograms of 1273. Can that be split and be multivalent? What's the potential for multivalency of the delivery system?
Right. So let me address the 100 microgram question. I mean, we've already announced that for booster in the future, we're going down to 50 micrograms. We had to choose that dose before we had the phase 2 data because we were doing the phase 1, phase 2, and phase 3 clinical trials in parallel. And we wanted to absolutely ensure efficacy. So we, went on the little bit higher side. But in terms of one of the true advantages of the mRNA platform is our ability to do multivalent vaccines. And we have a vaccine that's already in completing phase 2 clinical trials now for cytomegalovirus that has 6 different mRNAs in it. In this case, the 6 different mRNAs encode 6 different proteins from the virus, 5 of which have to make a multi-protein complex. And so making those multi-protein complexes are actually really difficult to do in a biologics context where you're making the protein outside the body and then trying to inject it. But coming back to the idea of multivalent vaccines, we're already testing in clinical trials now multivalent COVID vaccines where we have the original one plus one of the new variants. And we also are working on flu vaccines where we'd like to have multiple variants in there. And so the real question is, what is the maximum dose that you can give to have good tolerability? And then what is the minimum of each vaccine, of each mRNA to get good immunogenicity of the mRNA? And that's then tells you the limit of how many different mRNAs you can put into a single vaccine.
And I think it's a really important theme, this, because certainly in the UK, we've had basically 3 different vaccination drives. So the first was the flu shots, and then we've had the 2 COVID shots. And if you take COVID, it's the cost of deployment is about the same as the total cost of deployment of the COVID vaccines is about the same as the total cost of buying those vaccines. So to the extent we can take that down, ideally, to 1 single shot, which would be a booster shot combined with flu, presumably aimed just at the most vulnerable rather than everybody, but that's for the health experts to decide. Yeah. And that would massively improve the simplicity of and cost of deployment.
We completely agree with you, and so we're working toward that.
And completely feasible.
Yes.
Yep.
Yeah, and my point with the 100 micrograms is that it can tolerate at least that dose, whether slightly higher or not, and therefore how low can you go per antigen in a given delivery event. Yeah. And I didn't mean, Melissa, earlier to say how simple this technology is. I realize a ton of science has gone into allowing this to move forward quickly. The point that I was trying to make and want to push you a little bit on is the speed with which mRNA can be designed and deployed, which you've hinted at, in thinking about the flu vaccine. Our current way in which we make flu vaccine globally is a little bit clunky using eggs and millions of them. How the speed of this technology, when would you need to know the flu strain for this year to have enough time to have a vaccine ready for the fall? Currently it's months, it's 6 months or more in advance.
Well, I think that one of the things that the COVID crisis has caused us and other companies to do, Pfizer and BioNTech in particular, is to build out our manufacturing capacity in order to make millions and tens of millions, 100 millions of doses. Now that we have that capacity, then it should be rather quick to be able to make the vaccines. I don't want to commit us to any particular time frame because I think that's tough. But what I can say is that with our personalized cancer vaccines, which we make for one person, our typical turnaround time there is about 45 days. And so that would be, I think, the very short limit. But certainly it should be faster, much faster than the current flu vaccines.
It's on the order of 2-3 months versus 6 months, without committing you to a specific date, because I think manufacturing capacity and other issues, but the selection of the strains to put into the vaccine, in theory, could be delayed till later in the year and hopefully have a better match, in theory, to what ultimately circulates.
Absolutely. And that's part of the problem with the current flu vaccines is that you just don't have a good match to what is actually out there circulating.
I mean, there's a little bit of a gamble when the WHO selects those strains, correct? And then there's the assay development. And then, of course, for some manufacturers, Lindsey, you point out millions of eggs. It's hundreds of millions of eggs. And so, getting those eggs acquired and getting them delivered to where they need to be, and then literally having people stand over those eggs and inoculating each of those eggs, there just has to be a better way. And I think we also need to think about, the importance of getting this right. And I think this is where the messenger RNA platform is going to play a major role. We've now just gone through a season where there was relatively low exposure to the flu. So, it's going to be interesting to see what seasonal flu looks like when it does come back, when we're not masked, when people aren't wearing masks, aren't as vigilant about this. How important is it going to be to get these strains correct? And I think that's where the mRNA platform is going to play a big role.
And Ron, sorry, go ahead, Kate.
It goes back to valency. I mean, what is the limit to the number of different sequences you can actually put in? Because, I mean, at the moment, we've been talking about 2 or maybe 3, but, could it be 10? Could it be 12? And in which case, does it, do you actually need to tweak materially between seasons if you can capture the most?
And I can tell you that that's exactly that's research that's ongoing internal at Moderna. We are trying to push the envelope just as very far as we can to see what the maximum is.
And just along those lines, Ron, I mean, not only are there hundreds of millions of eggs and the delay in kinetics of selection, there's also egg-adapted mutations and other issues in serial passage. So this type of technology has certain advantages as we think about other vaccines that we globally deploy. How do you think about this for other gene delivery, AAV, other viral vector delivery systems? How do you, for gene therapy, how do you think of the mRNA technology compared to these other platforms and the potential on that side of the house?
Yeah, well, Kate just touched on it. I mean, if we're thinking about multivalency, you start to think about all these vaccines. When you start going into, start thinking about AAV vectors, you're thinking about much smaller cassette size, much smaller transcript size that are going into these AAVs. And I know that we've got dual vectors and we're starting to split them up and starting to look at things like that. But, on the lipid nanoparticle side, like Melissa said, we're doing the same thing, trying to push the limit there. And you start thinking about putting in double-digit KB sizes of transcript into these LNPs and still getting properly folded proteins, getting the right, when you look at the mass spec, they look exactly the way they're supposed to look like. I think it just opens the door wide open so that we don't need to be thinking about how we're going to fit these transcripts into the AAV, whether or not we're going to get them delivered to the nucleus. Here, you only have to get to the cytoplasm. You don't need to get incorporation. You don't need to worry about insertional mutagenesis. There's so many other things that come off the table with simply delivering messenger, and it's certainly not simple, but just getting to the cytoplasm versus delivering gene therapy. There are places for gene therapy. There are places for AAV. And we look at it very much like the central dogma of biology. Messenger RNA sits right in the middle, and I think the landscape is very wide open. But I think in the vaccine space, we're only seeing the tip of the iceberg for this.
Unfortunately, we've run out of time. There's so much more we could talk about. We've hit on such important topics that look to the future. Messenger RNA is the message, and we just have to decide what message we want to deliver to the cell for a salutary purpose. We've learned a tremendous amount in the past year about the potential, the widespread distribution, aspects of the safety. We still have much more to learn, but the promise of this technology, I think, could not be more front and center for all of us. So I'd like to thank all of you for your contributions to this discussion. And I will now like to introduce our next panel, which will be led by Dr. Nancy Berliner, who's Director of Clinical Research at the Brigham, and she will explore indications for GCT for hemophilia. But first, we have a few polling questions we'd like to answer. They should pop up on your screen now.